think I'll start it now. All right. All right. Hey, everybody. Thank you for joining in on the Marine Institute Graduate Society Seminar Series. We meet every week on Wednesdays at noon. Hopefully we can start having these things in person again soon, but I'll let you guys know. I know things are starting to open back up a bit more, but as far as I know, I think grad students are still encouraged to work from home. So I don't think we have much of a live audience. That's why I'm still keeping them online. This is being recorded and will be uploaded to our YouTube channel, where you can find most of all of our seminar series over the past couple of years. So I do want to take a second to recognize Dr. Jeffrey Hutchings, who recently passed. He gave a great seminar for us back in mid-January. He was a well-respected professor at Dalhousie and even recently wrote a book on ecology and evolution. He was also just, on a personal note, he was also just really kind to me, relating to me that he was a seminar coordinator at one point. So I really appreciated having that opportunity to speak with him. So I'm going to change gears now and provide the land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk, and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We'd also like to recognize the Inuit of Nunatsiavut and Nunatuckavut, and the Inuit of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So now please join me in welcoming DFO research scientist, Dr. Sarah Lehnert. Dr. Lehnert's studies primarily focus on salmon genetics and evolution. She graduated in 2016 with her Ph.D. from the University of Windsor, studying why king salmon are red. And not just why, but really why. She went on to do a postdoc working with Dr. Ian Bradbury. So in other words, she's also an expert on using incubation trays. We're really happy to have Dr. Lehnert with us today. And with that, I'll hand it over to her.
Thank you, Colin. So today I'm going to talk about some genomic tools that I've used to address evolutionary and applied questions. And I'm going to focus on a lot of the work that I've been doing on Pacific salmon, but also on Atlantic salmon. So just to give you a bit of background about myself and my research interests, I'm really interested in the variation that occurs in nature and the mechanisms that help promote that variation. So for example, we can have variation occurring at multiple different scales, including at the level of different species. But even within species, we can see differences between different populations when those populations are adapted to different environments. But even within the same population and in the same environment, individuals can still show differences. And those differences can be important for helping those individuals succeed in that same environment. And what I'm really interested in is looking at these population level differences, but also individual level differences within populations. So to give you an idea of some of the research that I've focused on through different projects, includes the type of variation such as alternative reproductive tactics like those found in Chinook salmon, where we see differences in male reproduction, including these large hook-nosed males compared to these small jack males. But both these males can use different strategies to obtain success. I've also looked at things such as color morphs in Chinook salmon, and I'll talk a lot about that today, as well as differences in immune gene diversity in the species and changes in these species associated with aquaculture, but also how aquaculture or stocking can influence other species like rainbow trout. And although a lot of my research focuses on salmon, I have worked on other species like green crab here, looking at the different invasion ecotypes that exist, as well as in how genetic variation can promote environmental adaptation, including in species like sea scallop. But more recently, my research has focused on Atlantic salmon. And things that I've been interested in lately include things such as chromosome level differences in the species, as well as genetic variation that can be important for promoting population persistence. So today I'm going to focus on some examples from salmon. And first I'll start off by talking about some of my research I did during my PhD. and this was done with the University of Windsor in Ontario. The title of my dissertation was Why Are Salmon Red? And one of the reasons that salmon are red is because of carotenoids that they get from their diet. Carotenoids are these red pigments that are deposited into the skin, eggs, and flesh of salmon. And it's thought that these carotenoids can be really beneficial to a salmon because they have many functions within the body. Carotenoids are powerful antioxidants, so they can help an animal deal with stress. They also make important in terms of immune function and vision, and they can also be important when it comes to sexual selection. So although many animals can use carotenoids and have benefits from carotenoids, salmon are the only group of fishes that actually have this red color flesh or muscle tissue that we see. So there's only a few species that deposit carotenoids into their flesh. So when we think about most of the fish we eat, most of them are white, cod, or halibut. So this begs the question, well, why are salmon red? So when we think of the evolution of the red flesh pigmentation trait, it's actually in the genera of salmonids, and that includes species like trout. This trait likely evolved in their common ancestor before these species started to diverge. And even though all these species exhibit flesh pigmentation, there's still a lot of variation that exists among these different species. For example, species like sockeye salmon have a much higher amount of carotenoids in their muscle tissue, whereas like Atlantic salmon or pink salmon or char have much lower levels compared to sockeye. We also see variation within species in the level of carotenoid pigmentation. So this could suggest that there's adaptation associated with carotenoids that could benefit salmon in terms of the biotic or abiotic factors that they encounter. And this suggests that there could be costs and benefits associated with this trait. What we know that this trait's only evolved in a small group of fishes, so perhaps there are costs associated with it. Otherwise, why aren't more fish showing this red flesh pigmentation? And we also see variation among these different species, suggesting that there could be additional costs within this group of fish, but there are likely also benefits as well. And I was interested in trying to understand these benefits and costs. So to do this, I used a study system of Chinook salmon. Chinook salmon are unique among salmonids because they exhibit two different color morphs. There's the red Chinook salmon, which are kind of like the normal salmon that we're used to. They have red flesh, they have red eggs, and they have red skin during spawning. But there's also a white Chinook salmon morph that exists, and this white morph has white flesh, it has light colored eggs, and its skin turns a gray color during spawning. So a lot of people might assume that this is controlled by their diet, but it's actually controlled by their genetics. So no matter how many carotenoids a white salmon eats, it can never become red. And what's interesting about these two morphs is that they occur at different frequencies in different populations. So for example, some rivers are 100% red salmon, some are 100% white, and other rivers have 50-50. So I'll talk about one of those rivers in particular, which is the Quesnel River system in British Columbia. The Quesnel River, as I said, has about 50% red Chinook salmon and 50% white Chinook salmon. So it presents a unique opportunity to be able to study these two morphs and look at how carotenoids influence them. And this river system is located as part of the Fraser River system. So Vancouver's down here, and that's where the Fraser goes in and moves up into the interior of BC. And here you have the Quesnel River system. So this is where we're working. And what I was interested in is looking at how carotenoids influence fitness in Chinook salmon throughout their entire life cycle. But today I want to focus on talking about some of the fitness consequences of carotenoids, or the trade-offs that can exist at the egg stage. So as I mentioned, these eggs from Chinook, from red and white Chinook, can look very different. For example, the red eggs are much redder in color, whereas the eggs of a white female are much paler in color. Now, this doesn't just result in visual differences, but we can find measurable differences in the carotenoid content of their eggs as well. So when we look at the eggs of red females, on average, they have about 6 times more carotenoids than the eggs of white females. And we can see that they also show a greater amount of variation in carotenoid content as well, whereas white females have generally very low levels of carotenoids in their eggs. So this could potentially lead to differences in terms of survival. And the reason that I say that is because a lot of fish or other species put carotenoids into their eggs, and it's thought to be beneficial in terms of survival. So even species like birds, like chickens, put carotenoids into their eggs, and that's why they have that yellow yolk in their eggs. So it's thought that during this really period of rapid embryonic growth, these carotenoids could be important for protecting an embryo against oxidative stress. So that could lead to higher survival if you have more carotenoids. But it's also possible that because these eggs are really different in color, they also might experience differences in predation risk. Red eggs might be more noticeable to a predator. So I was interested in looking at these questions in Chinook salmon, and we used a common garden design to look at multiple questions from the fertilization stage up to the smolt stage when salmon were getting ready to migrate to the ocean. So to give you an idea of what this research looked like, here we are in the field in the Quesnel River, where we can set a net across the river here. And we use a boat where the net's tied off at shore, and the net's released from the boat and quickly drifts to shore, catching any salmon that are in the area. Once the salmon arrive on shore in the net, we quickly untangle them and we transport them back to the Quesnel River Research Center, which is owned by the University of Northern British Columbia. Then we move the salmon into spawning channels so they can get ready for spawning. And when they're ready for spawning, we collect their gametes, including the sperm and the eggs, and we can perform fertilizations and start raising those eggs in incubation trays and monitor their success across time. So the first thing that we did was we looked at the survival of the eggs from the fertilization stage to the eyed egg stage. And the eyed egg stage represents the stage where you can actually start to see the little embryo in the egg and see the little eyes on the salmon. So at this stage, what we found was that there was no significant difference in egg survival between the eggs of red females and the eggs of white females. So despite those really strong differences in carotenoid content, we saw no difference in survival. But the next question that we had was, well, maybe within one of these morphs, we can see associations with carotenoids. So we know that the red females have a lot of variation in carotenoids. Perhaps within this morph, there's associations with survival. So this is what we looked at next. When we looked at the egg carotenoid content and the survival of the individuals, we found that red females had a significant relationship between survival and carotenoids. Higher carotenoids led to higher incubation success. But we did not see the same pattern within the white Chinook salmon morph. Even females with really high levels of, or really high survival had really low levels of carotenoids. So it's possible that this white morph has evolved some way to compensate for their reduced carotenoids. Based on the relationship that we see in the red morph, we'd expect white Chinook salmon should have very low survival, but they don't. So maybe they invest other things into their eggs, such as other antioxidants that can potentially help improve survival in that morph. Now, the next question that we had related to predation. This is a picture of a Chinook salmon female, and she's getting ready to spawn. Next to her is a male Chinook salmon, but you also might notice some other fish species here, and that includes rainbow trout. Once the female starts depositing her eggs, this trout will come in and start to eat her eggs. So rainbow trout represent an ecologically relevant predator to Chinook salmon eggs, but there are a number of other predators on salmon eggs as well. But I'm going to focus on rainbow trout here today. So I was interested in finding out whether different eggs might experience different predation risk. So for this experiment, we used choice trials with video recordings. And what we did is we simultaneously released a red egg and a white egg into a tank with rainbow trout. Within the tank, we recorded which egg was eaten first and how much time it took to eat each of these eggs. This was replicated with multiple tanks and multiple populations and across multiple days to measure which of these eggs were being consumed more often. So before I get into the results of this, I will say again that we did use different populations of rainbow trout. So in this experiment, we had one called Blackwater, which comes from a river that is located close to the Quesnel River. So these rainbow trout might have experience with red and white Chinook salmon eggs. The other river system that we use is the Pennask, which doesn't have a spawning population of Chinook salmon, so they wouldn't have experience with red or white eggs or any experience with Chinook salmon eggs in general. So it's possible these populations might show differences. But what we found in our experiment was that red eggs were eaten first significantly more of the time than white eggs. And this was true in both populations of rainbow trout. In addition, when we looked at how long it took to eat the eggs, red eggs were consumed significantly faster compared to white eggs, and there was no difference between the populations. So overall, this suggests that there are costs associated with carotenoids, and this is the first time that that's been documented in salmon. So overall, in the early life stages, we could show trade-offs that exist in terms of carotenoids. We show that carotenoids can increase survival during incubation, at least within the red morph, but higher levels of carotenoids can also lead to greater predation risk. So that is an important trade-off that exists at the egg stage. Through my PhD, we also looked at a number of other traits throughout the life cycle, and I won't go into details about these, but I'll just try to summarize some of those findings here. We also looked at the juvenile stage, looking at growth and survival, but we did not document any differences between the offspring of red and white salmon. We also looked at things such as immune function. We challenged these fish with a virus, but we found no differences in their ability to respond. But we did find differences in the population when we looked at genetic differences in their immune genes. We found differences in the types of alleles that they had and the diversity of those. In addition, we looked at mate choice in this system and we found evidence that there is some assortative mating in the population based on color, where red females and white females bias paternity of their eggs in favor of a male that's the same color as themselves. But they can do this in different ways, either through pre or post-copulatory processes. So that summarizes some of the trade-offs that can exist, or maybe some of the reasons that red salmon exists, but also why white salmon could also exist. But I want to now talk about the genetics of this trait. So I'm interested in this from an evolutionary perspective, but this trait's also important from an economic perspective. And that's because aquaculture producers often want to have a redder salmon product. And that's because consumers like ourselves will associate the color of a salmon with the quality of the salmon. So aquaculture producers can invest money in carotenoids, but carotenoids are expensive. And even when you feed your salmon a lot of carotenoids, there can still be variation in color. This is also a difficult trait to use for selective breeding. So this could be a good trait that we could look at the genetic markers for this trait and then breed salmon based on those genetic markers to improve color of the salmon. So this work was carried out in collaboration with DFO, but it was also done in collaboration with Yellow Island Aquaculture, which is a small organic salmon farm located off of Quadra Island in BC. And this is what the view looks like from the salmon farm. And a lot of my master's and PhD research was conducted in collaboration with Yellow Island. So what we wanted to do for this project was create a family of salmon that had a variety of different colors. So to do this, we used an F2 back cross design, and I'll explain what that means. We had a red salmon and a white salmon that were mated together to produce an F1 offspring. This salmon would carry copies of the red and the white alleles, and this salmon was mated with a white salmon. And that resulted in a family of fish that had a variety of different flesh colors. So within this family, we could then look at each fish and score them based on their phenotype. So we cut them open and looked at what color they were and gave them a color score. And then we also looked at their genotype. So we took a genetic sample and we looked at thousands of markers across the genome. And then what we were interested in doing is looking at which of those genetic markers were associated with the color of the salmon. So we did this, and this kind of summarizes our main findings, but this is what we call a Manhattan plot, and I'll explain what it represents. Along the x-axis is all the chromosomes of Chinook salmon, and each dot here represents a different genetic marker where their position is along each chromosome. Axis, we have the significance of those genetic markers with higher values indicating more significant. And what you might notice is that on chromosome 30, you have a whole bunch of markers that are highly significant, with the most significant one being this one right here. When we start to look at the association with color for this marker, we see that there are two genotypes present, and there are significant differences in the color of these salmon based on their genotype. So for example, these ones with the GT genotype are significantly redder in color compared to these salmon over here with the GG genotype. So these two genotypes produce salmon that are quite different in color, and this suggests that this genetic marker is probably located quite close to the gene that influences color in salmon. So what gene is closest to this genetic marker? Well, when we look at this in more detail, we have our marker right here, and beside it is a gene called beta-carotene oxygenase 2-like gene, or BCO2. This gene is involved in carotenoid cleaving, so it has a direct role that could influence carotenoid pigmentation in salmon. And it's actually been found to be associated with carotenoid accumulation in other animals, such as in mammals and lizards and birds. So it's a good candidate gene for explaining the differences in pigmentation that we see. Now, this gene encodes an enzyme that's responsible for breaking down carotenoid pigments. So we looked at the expression of this gene in salmon that were red and white, and what we found was that individuals that were expressing this gene at a higher rate were those that were white. And that's because in this group of fish, those carotenoids are getting broken down into colorless derivatives. So that results in them being white and not accumulating carotenoids in their tissue. But when we disrupt the function of this gene or reduce the expression of it, that results in those carotenoids not being broken down, and instead they start accumulating in the salmon, and that results in that bright red color that we see. So differences in expression of this gene can explain the differences between red and white salmon. So if we go back to this larger question of why salmon are red, well, one of the answers is because of a mutation that happened in this beta-carotene oxygenase 2-like gene. This disruption of this gene that reduced the expression of it allowed salmon to start accumulating carotenoids in their tissues. And we estimate that this happened about 30 million years ago in a common ancestor of salmon as these species were starting to diverge from each other. This resulted in this important fitness-related trait that could influence a variety of different factors in the life cycle of the salmon, including the incubation survival, the predation risk, their mating system, as well as immune function. And it's hypothesized that this also potentially led to the evolution of anadromy in salmonids. And this is because during this migration that salmon undertake to fresh water, Those carotenoids are found in the muscle tissue, but as they start moving upstream and into these river systems, they start moving those carotenoids out of their flesh and into their bloodstream. And this can help promote the maintenance of the salmon during that stressful time period. So it's possible that this could have been important for helping those fish undertake those long migrations. So overall, with my PhD project, we answered a number of questions that help explain some of the benefits of carotenoids, but also the costs. But I think this research has also led to a lot of other questions as well. But I'm going to stop there, and I'm going to start focusing on a species that might be more familiar to people, which is the Atlantic salmon. Atlantic salmon are found across the North Atlantic Ocean, and when they're during the freshwater stage, they occupy rivers in both North America as well as throughout Europe. Their genetic structure we see is structured at multiple different spatial scales, but the biggest genetic difference in this species comes between the two continents. So European salmon and North American salmon are genetically quite different. They have been isolated for about 600,000 years, and they even have differences in their chromosomes. And although they are considered the same species, there is some evidence that there's reproductive incompatibilities between these two groups. So some of the research that I did during my postdoc with Ian Bradbury at DFO focused on looking at these differences between European and North American salmon. When we sample populations across Canada, including from Newfoundland and Labrador, as well as populations in Europe, mostly from Norway, we can see in this PCA here that these populations are clearly genetically distinct, where there's quite a bit of separation between them. And we see this being true across the entire genome of these salmon, where this measure of genetic divergence, FST, is quite high, estimated around 26% on average, which is quite high for a group for populations of the same species. When we looked at what genes were associated with this difference, we found that there are many genes throughout the genome that showed really high levels of differentiation, and these were associated with the enrichment of specific processes that could potentially highlight some differences, adaptive differences between the two continents, potentially related to ocean navigation and immunity. So I've just told you that there's really big differences between North American and European salmon. But despite these really large differences, there have been opportunities for these salmon to interbreed. And these interbreeding events are known as secondary contact events. And this has occurred between European and North American salmon towards the end of the last glaciation. So this map here shows what it looked like here. This is Newfoundland under this ice sheet at the end of the last glacial period when ice extent was still at its highest. And as time went on, after about 20,000 years ago, these ice sheets started to retreat. And then we had these habitats starting to open up. So when these areas were covered in glaciers, salmon were existing in a southern refuge in the south here. But as these environments started to open up, salmon started to recolonize different river systems from the south to the north. So this map shows what it looked like about 12,000 years ago. At this time, you can see that a lot of Newfoundland was still covered in glacier, but parts of it were starting to open up and providing new habitat for salmon to colonize. So at this time, it's thought that as North American salmon were recolonizing these rivers, there was also opportunity for European salmon to attempt to colonize these rivers as well, specifically in certain areas of Canada. And what we know, we do know where those areas are located based on some mitochondrial evidence. The mitochondrial markers can be useful for looking at these historical lineages of Atlantic salmon, looking at the difference between North America and Europe. When this has been investigated in European populations, it's often found that most salmon in Europe show this European lineage, which is indicated by red. But once we look at more northern regions, there is some evidence of North American salmon interbreeding up here in the north. If we look at North America, we can see that there's evidence that most of these southern populations are North American in origin. But as we move more northward, like in Labrador, we see evidence of this European lineage, suggesting European introgression. Specifically, if we look at the island of Newfoundland, it's really interesting because there are a number of populations across Newfoundland that have a large proportion of European introgression. And these are localized often along the Avalon Peninsula, but also throughout other regions of the island. So for my postdoc work, I was interested in looking at this further using the whole genome of the salmon. So we wanted to look across all genetic markers that we sampled to see if there were specific parts of the genome with greater introgression, and what parts of the genome that had that signal. So when we looked at this, we did find specific regions that showed evidence of European DNA here in North America. And what we see from this map is that the blue represents the European signal in the DNA, but the red represents North American. When we look at southern populations in North America, we often see that they're mostly showing that North American signal. But as we move north, like into southern Newfoundland, as well as places in Labrador, especially Lake Melville, we see a higher proportion of this European expression. So what this is actually associated with is specific chromosomes in the genome. So we don't see it across the whole genome, but we see it localized to specific regions like chromosome 1 and chromosome 23. And these two chromosomes are associated with a chromosome rearrangement. So I'll try to explain what that means. Between Europe and North America, salmon have different numbers of chromosomes, and this is due to different arrangements of their chromosomes. So chromosome 1 and 23 looks like this in Europe, where there are 2 arms of chromosome 1 and 1 arm of chromosome 23. But when we come over here to North America, what we see is that one of these chromosome arms has broken off and attached to chromosome 23. This results in a different chromosome rearrangement. This is known as a translocation. But as you can see, this doesn't change how many chromosomes they have. It still results in 2 different chromosomes. But some other arrangements can influence how many chromosomes salmon have, which is why North American salmon have a reduction in chromosomes because of other rearrangements. So hopefully that all made sense. But what we've been finding through this research is that in Newfoundland and Labrador, some salmon carry copies of this European type chromosome, or other salmon carry copies of the North American type. And there are even some salmon that carry a copy of both of these types. And what we're finding in this region of the genome is that there's evidence of selection acting on these chromosomes, which could potentially suggest a role for adaptation. So the reason that I bring this up is because chromosome rearrangements have been identified in a number of different species recently, as we get better and better genomic tools to start to look at this. And this is true for a lot of species here on the East Coast, including species like Atlantic cod, rainbow trout, Atlantic herring, Atlantic halibut, and capelin. All these species show evidence of a chromosome rearrangement, and in many of these cases, those rearrangements are directly involved in adaptation. So it's possible that this rearrangement that we see in salmon could be important for adaptation to different environments or life history. And this is something that a graduate student at Dalhousie looked at in more detail. So Beth Watson was a master's student with Ian Bradbury and Paul Benson, and through her work, she looked at this chromosome rearrangement in more detail. And she looked at it in Placentia Bay here in southern Newfoundland. And what she found was that she found a higher proportion of that European type chromosome over here along the Avalon. And it's possible that this could reflect adaptation. So to investigate that question of adaptation, Beth looked at which environmental variables were associated with it. And what she found was that there was evidence for potential adaptation because this rearrangement was associated with temperature and climate. So for example, when she looked at the proportion of the North American type chromosome, she found that it was highly associated with her temperature variable. And on the map here, it kind of highlights that, where the different size circles represents the frequency of the North American type, and the colors in the map represent annual temperature range. So what Best concluded was this European type chromosome was associated with less temperature variability and warmer winter temperatures. And it's important that we understand that what types of variation exist in salmon and how they relate to adaptation, because this can be important when we predict responses to future changes. So that leads me kind of into the next part of this talk, where I want to focus on the threats facing Atlantic salmon. So Atlantic salmon, as many people probably know, have been experiencing declines in recent decades, including extirpations in some populations. There are various factors that can influence and threaten salmon populations, and some that have been identified include things such as the impacts from aquaculture, historical fishing pressure, habitat alteration, and climate change. But often the specific causes of population decline remain unknown. And one issue that we face is we can only monitor so many populations directly using traditional methods. So another way that we could start to look at how populations are changing across time is using genomic tools. And I'll talk about one method that we can use to track how populations change across time. This includes some new methods that use genomics to reconstruct effective population size across time. Now, effective population size doesn't necessarily equate exactly to the census size of the population, but it gives a good idea for the diversity and of the population. So with DNA samples that we collect today, we can reconstruct samples that reconstruct how populations have changed in the past. So for example, we can collect samples now and reconstruct what their population size was back many generations ago. And this method, I won't go into details of exactly how it works, but it's based on genetic linkage information and recombination. And this can tell us about what effective population size is today, but also what it was at different, but also what it past. And we use this method to track population changes in Atlantic salmon. And with this method, we focused on recent decades, and we looked at which populations had shown significant declines across that time period. And what we found was that about 60% of populations in Europe and in North America experienced significant declines during that time period. And again, these represent declines in effective population size. And these declining populations are indicated by the red dots on the map. What we wanted to do next was try to understand what kind of factors might be associated with these declines. So we used random forest classification to look at a bunch of different factors to help explain the variation in declines. So this includes things such as climate, variables like temperature and precipitation, as well as anthropogenic factors such as human density and aquaculture. And what we found when we looked at this in North America is that variables that were important in the model included things such as the ones at the top here, which include things like habitat alteration, which we used human density as a proxy for, and also things such as climate variables like temperature. So at this broad spatial scale of the continent, I will say that these could only explain a small portion of the variation in declines. And this is something that Jeff Hutchings said a few weeks ago, where he said there's a need to focus on local scale stressors that can affect populations in different ways. So for example, at local scales, there might be factors that are not the same at a large spatial scale. And this can include things such as different pressures from predation or aquaculture, how low abundance might influence these populations' ability to respond to change, and also how climate change might impact these populations. So I want to focus on this question of climate change in relation to Atlantic salmon. We can use genomic methods to start to predict how populations might respond to climate change. And this is being done in a number of different species where basically what you're doing is looking at what genes or genetic markers are associated with climate, and then looking at which populations might be most vulnerable to climate in the future. And you do this by looking at which populations will have to change the most genetically to be able to adapt to future changes in climate. And this kind of work has been done in species like the yellow warbler here, where the redder parts of the map indicate populations that are more vulnerable to climate change. And in this study, what they found was that those populations were already showing evidence of declines, which could all indicate their vulnerability. In addition, this type of work has been done in species like Arctic char here in Newfoundland and Labrador, where species or populations in the south were found to be more vulnerable to climate change. So I'm really interested in this type of work for Atlantic salmon. And some of the research that we've done so far, which is preliminary, suggests that populations in Newfoundland might be more vulnerable to climate change. But we really need to do more work to really understand climate adaptation in this species. We know that genetics and environment can influence the ability of salmon to respond to temperature stress. So it's possible that different populations will have to respond in different ways. So I would like to focus my research on looking at the genetic markers that are associated with climate factors, but also using lab-based experiments to really dig into which genes are responsible for that climate adaptation. And that can help understand the differences among populations here in Newfoundland and help better forecast how they might change under climate change. So with that, I'd just like to conclude today by talking a little bit about how we've applied this research. So with genomics, we can help. I hope that I've convinced you that we can answer lots of evolutionary and applied questions. These can help us identify mechanisms that help maintain variation in nature. It can also help us identify which genes are responsible for adaptation, but also things like chromosome rearrangements, how they can contribute to adaptation as well. We can also use genomics to help understand how populations have been changing, but also forecast how they might change in the future. And overall, these tools can contribute to species management and conservation. And a lot of the work that I've been doing recently applies these genetic tools to conservation of Atlantic salmon by helping with the reassessment of Atlantic salmon for Kasiwik. So with that, I'll just end today by acknowledging all the people that have helped me through different parts of my research, from my PhD and postdoc and now as a scientist here at DFO. And I would be happy to take any questions that you guys might have.
All right, thank you so much, Sarah. You have a great pace. I was able to keep up with you. That was fantastic. Thank you so much.
Yeah, no worries.
Yeah, do we have any questions for Sarah? You can go ahead and raise your hand, or yeah, and I'll call on you. So I do have a question. Coming from the Pacific coast, how much do you think single kill events affect your results on genetic diversity around Newfoundland and Labrador? Like, I'm thinking of like when just not too long ago in BC, like that canyon, and it blocked the river, and they just, they were salmon, whatever, they're trying to figure out how to get the salmon across the Rocky Dam. But yeah, anyway, look, how much do you think that those big single kill events might affect your results when you're studying stream by stream the genetic diversity?
Yeah, so are you talking like the mortality events related to temperature that have been happening recently,
Maybe,
or?
maybe not just mortality, but I mean not temperature, but I'm just thinking of maybe so you showed the map of the genetic diversity of those little the pie charts showing how much of the European genes that they have, some of these Atlantic salmon have in Newfoundland. And I was just wondering, and you were making a correlation with that, right? But I was just wondering how much uh, um, extirpation of a specific population, maybe not the entire stream, but maybe a certain run, um, might influence, the lack of that lost genetic diversity. Have you thought about that, or does that really factor in anything over here on this side of the North America?
Yeah, that's a good question. I'm not sure, but I guess it could be some historical events that had happened as those populations were trying to colonize those rivers. Might have influenced which individuals were successful. Um, and yeah, I'm not sure that I can answer that
It's
very
okay.
well.
How much? How
But
much?
I mean,
How much? Uh, s- uh, what is it called when salmon go from one stream to another? I forget what it's called, but um,
straying.
yeah, straying. How much straying is there in Atlantic salmon compared to the Pacific salmons?
Yeah, so straying is estimated to be around 6%. So it might be kind of similar to Chinook salmon. And straying is expected to be spatially limited, where they're going to go to nearby rivers, not to really distant ones, usually within about 60 kilometers. And even when they do stray, there might be selection happening that might prevent those individuals from being successful when they stray.
Sure. Okay. Um, and then since nobody else is asking questions, I do have another one. Uh, do you think that you can predict? Do you think the association between the color of the eggs um, and their ability, how fast they're eaten, or the survivability, I guess? Uh, do you know? Do you know if you could predict how successful they might be if you know the predators that are in the area or the population? Like I'm thinking of river enrichment. When you use a hatchery to help restock a river, do you want to take into account the color of the egg, that kind of stuff?
Yeah, that's a good question. It's definitely something that they should consider, although um, I guess it would depend on the river itself. So we were looking at one population and within like experimental tanks. So it's totally possible that maybe different river systems could promote higher predation risk on red eggs, or maybe in some rivers it's negligible, the difference between red and white eggs, depending on the environmental conditions, like the turbidity of the river or something like that. So it's definitely something that we could look at a larger scale, and it would be important to consider what type of egg you're putting into the environment, depending on what the conditions were.
Yeah, that's the first thing I thought about. Was I know they really try hard to improve genetic diversity in those hatcheries, but maybe that's not something they ever thought about, was how much
Mhm.
carotenoids, or anything like that.
Yeah.
All right, I think that might be it. So we have a few more minutes though, if anybody has any questions. I am going to just say though, uh, before I end it, I do want to introduce next week's seminar. We didn't have anyone booked officially, so I'm just going to give my seminar again. It's on animal identification. So when I gave it in the fall, we didn't stream or record it. So if you need some identification tips for stomach content analysis, myofaunal, epifaunal, or pelagic samples that you're doing, or if you just want to know how to identify that super cool sandhopper, amphipod, jellyfish, snail, or anything, just come hang out and listen to me ramble on about animal identification. So that's vertebrates and invertebrates, marine. And if you have something specific that you want me to cover, shoot me or MIGS an email, and I'll be sure to cover it, including the less familiar things that wouldn't otherwise get touched on for time's sake, like zooplankton, bryozoans, and stuff like that. So anyway, I did that for like 4 years, so so I can talk about that kind of stuff. Oh, it looks like we have a question. Tanya.
Hi, yeah. I am just going out on a limb here. So I thought it was really cool. It kind of took me back to my master's where I did a lot of work with the Chilco and Gates Creek sockeye salmon, looking at heart rate and reproductive fitness. And I was sort of wondering if there was any studies looking at, because you're talking about these different dynamic types and how it can relate to predation. I was wondering if there's any relation to physiological performance that was done, because we see a lot of variability in different stocks of sockeye salmon in terms of heart rate performance and how that translates to temperature tolerances, thermal tolerances, and reproductive performance. So yeah, I was just kind of curious because I've never seen I actually didn't know about the red and white different subpopulations, so that was kind of cool. So yeah,
it's a good question. So unfortunately, there hasn't actually been that much research on the red and the white Chinook salmon. There was some earlier research in the late 80s, early 90s, and then nobody has really been looking at it much until some of the research that we've done in Windsor more recently. But yeah, that has not been done, but it's a good question. And I think a lot of important questions are still left to be answered that could relate to their ability to perform these long migrations. It had been thought that white salmon were mostly located in like more coastal environments, but really there are white Chinook salmon that exists at, very far up the Fraser River. So that it doesn't seem to prevent them from being able to undertake those long migrations, but there might be differences that they gain somehow in terms of having that reduction in carotenoids. Maybe they find mechanisms to compensate for that. But it definitely would be an interesting area to look more at, their migration capacity in terms of carotenoids for sure.
Yeah, great, thanks. I hadn't heard of anything, so that kind of makes sense. But it is cool. Thank you very much.
Yeah.
I think that's it for questions. Yeah, so thank you so much, Dr. Lehnert, for joining us today and giving us that great seminar.
Thank you.
I'll end it there. Bye.